Hi, I'm Natalie. And I'm Rebecca, and we're from Prostate for Life. ERG is a member of a group of genes involved in the regulation of a range of essential cell properties. It is not normally expressed in the cells that line the prostate. However, in 50% of prostate cancers, a genetic alteration occurs whereby an androgen-responsive promoter, TMPRSS2, fuses with the transcription factor, ERG. This results in the aberrant expression of ERG, which activates the expression of ZEB1 and ZEB2. This causes prostate cancer cells to lose E-cadherin, which leads to the loss of cell adhesion, enabling prostate cancer cells to acquire an epithelial to mesenchymal transition. ERG also activates the Wnt pathway through FZD4 to induce further loss of cell adhesion, allowing for prostate cancer cells to gain migratory and invasive characteristics. This activation of invasion and metastasis is a hallmark of cancer. Given the prevalence and aggressiveness of ERG-positive prostate cancers, an effective treatment is indisputably required. We have combined a pre-existing targeting mechanism to accelerate the turnover rate of all pre-existing mutant ERG proteins, and one of our omics targets the fused ERG gene, in order to 1. Prevent the transcription process and therefore formation of these mutant ERG proteins, and 2. Eliminate these proteins that are already existing. We call it CRISPR-10. The first part of CRISPR-10 involves CRISPR interference. This regulates gene expression mainly on the transcriptional level. It does this by working independently of host cellular mechanisms to silence a target gene with up to 99.9% repression. CRISPRi requires only a nuclease-deactivated Cas9 protein, which lacks endonucleolytic activity, paired with a single guide RNA. The single guide RNA is designed to consist of 20 base pairs that are complementary to the target gene. In this case, the TMPRSS2 gene. The single guide RNA will be designed so that it will consist of 20 base pairs that are complementary to the promoter region of the non-template strand in the fusion gene. Targeting the promoter region will sterically prevent the association between key cis-acting DNA motifs and their cognate trans-acting transcription factors, resulting in the repression of transcriptional initiation. It is important that the sgRNA is complementary to the non-template strand because it will more strongly repress transcription compared to being complementary to the template strand. The second part of our treatment will involve a one-off microinjection of degrazin into the prostate. This will target the pre-existing mutant ERG proteins to accelerate their turnover rate, decreasing their ability to contribute to prostate cancer metastasis. The stability of ERG proteins in the prostate are modulated by the interplay of ubiquitination and debuquitination. Ubiquitin goes through a series of ATP-dependent enzymatic steps. This involves E1, a ubiquitin activating enzyme, E2, a ubiquitin conjugating enzyme, and E3, which is a ubiquitin ligase, in the prostate known as TRIM25. TRIM25 binds to the ERG protein and to an E2 to form an E2-E3 complex. After a series of cycles, ubiquitin becomes covalently linked to the ERG protein as polyubiquitin chains. This tags the protein and allows it to be degraded by the proteasome. However, this process is reversed by deubiquitinating enzymes. In the prostate, USP9X is a deubiquitinating enzyme that cleaves the covalent ubiquitin bonds between ubiquitin and the ERG protein, as well as between the ubiquitin molecules in a chain. This prevents the protein from being degraded and hence stabilizes the ERG protein. The candidate degrazin is a small molecule that is a selective deubiquitinase inhibitor that will inhibit USP9X activity. By doing this accelerates the turnover rate of ERG by allowing it to be degraded and hence efficiently removes ERG. Our treatment is particularly beneficial because we are specifically targeting the prostate by targeting a prostate-specific gene, TMPRSS2 CRISPR-10 is also minimally invasive. The first step in eliminating prostate cancer altogether is by eliminating the worst forms of it, and CRISPR-10 does just that. By contributing financially to the development of CRISPR-10, your corporate social responsibility will be directly increased. Given the plausibility of the mechanisms behind CRISPR-10, the risks involved in further research are limited and is an extremely worthwhile investment.